In this video, we will discuss about alpha-gal-epitope. We have understand some of the information about alpha-gal-epitope in one of our previous videos, and we will continue our discussion on alpha-gal-epitope and know some more information about this. So far, we have understood that alpha-gal-epitope is absent in human beings at present, uh, and from the human genome sequence, it has been predicted that in the past this alpha-gal epitope might be active. However, because of the evolution, or this alpha-gal epitope has enzyme become inactive to that catalyzes the formation of alpha-gal epitope. And with time, the anti-alpha-gal epitope, there is a spelling mistake. The anti-alpha-gal antibody is present in the human beings. So whenever a alpha-gal epitope comes in contact with human beings or human cells, it will lead to immune response, and that is the reason why when a organs from alpha gal positive donor has been transplanted to alpha gal negative donor, it will lead to hyperacute rejection because of the presence of anti-gal antibody inside the alpha gal negative donor, especially in human beings. And the enzyme that catalyzes this alpha gal epitope is alpha gal transferase enzyme that is usually inactive in human at present, in present time. However, in the past, it it can be active based on the human genome sequencing. And there is and here is the example of alpha-gal epitope formation in the mouse. This is one of the way by which alpha-gal epitope form. However, there are other ways also for the formation of alpha-gal epitope in the mouse. And this alpha-galactosyl transferase is present in the Golgi apparatus that leads to formation of alpha-gal epitope. So how this happens is that the N-acetyl galactosamine uh, glycan when comes in Golgi, it will when comes in Golgi, this UDP containing galactose, uh, UDP containing alpha gal epitope will be transferred to the N-acetyl galactosamine with the help of alpha 1,3 galactosyl transferase. So in this way, alpha gal epitope is been formed in the mouse, and this is just one of the way by which alpha gal epitope form. There are other ways also, and this is the study that has been conducted to understand the differences in the alpha galactosyl pseudogene and the alpha 1,3 galactosyl transferase gene that are present in other organisms alpha gal where alpha 1,3 galactosyl transferase is active. So, and this pseudogene has been formed from the human genes that might lead to frame shift of the nucleotides in a way that the final enzyme that is produced is inactive. And here is the some of the sequence that are being identified that are different from other alpha galactosyl transferase present in other organism. Here we can see that the nucleotide CTG is different from the TCA nucleotide of Machia. However, it is similar to second and third. And in this case, also it is similar to the first case where the nucleotide is different from second, but it is similar to third and fourth. Whereas in this case, we can see that nucleotide is different with the third and second. However, it is similar to the fourth. Uh, and there is the also the case where we can see that the nucleotide is different from in first and fourth. How first and fourth, however, it is similar to first and third. So there is always a point where there is a mismatch of nucleotide of human inactive alpha galactosyl transferase pseudogene and the alpha galactosyl transferase pseudogene that are active in different organisms. And this is the one of the application of the information that has been gathered about alpha gal epitope. In this case, when people are not aware or about the alpha gal epitope, then organs from pig, when it is generally a alpha gal positive donor. It when the organ from pig is been transferred to the human, which contains anti-alpha gal epitope antibodies. So this might lead to rejection because of hyperacute immune response. Hyperacute immune response because uh, the pig is a alpha gal positive donor. However, when the pig has been modified with the knockout of alpha 1,3 galactosyl transferase gene, then it is no longer have alpha 1,3 alpha 1,3 gal epitope. So organ from this kind of pig, mutated pig, can be transplanted to the humans, and it will not leads to hyperacute rejection. And here it is a misspelling. It should be alpha gal 1,3 gal negative. This is another example by which alpha gal epitope can be used to treat cancer. In this case, cancer cell have been modified with alpha gal epitope. So that can be easily recognized by antigen presenting cell, and it will gives a signal to the T cell to generate an immune response against the cells that contain alpha gal epitope. That is, in this case, is cancer cell. So in this case, people can target only the cancer cell. In this case, this technology can target only the cancer cell without affecting the healthy cells. So this is one of the strategy uh, that can be utilized to treat cancer cell. However, it is still uh, this strategy is still not been successfully commercialized or successfully executed, and a lot of optimization still needs to be done. However, the idea the alpha gal epitope information provides is promising. Thank you for your time. Have a good day.